Okay, hello again. I know we're all still grabbing lunch, but before we get started, I just wanted to make a quick announcement regarding next week's seminar, because next week's seminar is in an unusual place and has a slightly unusual time. So next week, we will have Damien DeFontaine by Zoom. 12:30 is going to be the actual talk, but the actual talk, so just like usual, but the actual talk is only going to be half an hour, and we'll be meeting in TCS 358, and I'll send a Canvas update just to make sure that's all good. And we're sort of doing an unusual time to just sort of accommodate time zones. So before that, from 12 to 12:30, we're basically going to front load the Q&A part before the talk. So if you are free from 12 to 12:30, there will be an optional Q&A with Damien before the talk, and then he'll give his actual talk from 12:30 to 1 at the usual time. So I will send a Canvas update just to sort of remind everybody about that. I think I maybe sent something about this earlier in the semester, but now that we're almost here, next week is going to be weird. Okay, so any questions about that? Although I'll also send a reminder since I know that was a lot of info very quickly. Okay. Okay, so that's all for next week. So for this week, we are very pleased to have Professor William Yu, who is a professor here at CMU in Comp Bio. William is a computational biologist and an applied mathematician. He's in compression, genomics, privacy, and sketching. And before he came to CMU, he was an assistant professor in math at the University of Toronto. Before that, he was a grad student with Bonnie Berger in math at MIT and a postdoc with Griffin Weber in the biomedical informatics at Harvard Medical School. So you're here to tell us all about genomics and privacy and medical privacy and all sorts of other things. So
thanks so much, Sarah. Can everyone hear me? Okay, yeah. So I am happy if anyone has questions during the talk. That is useful because this talk was originally calibrated for our Comp Bio Masters program. And so some of the privacy and security stuff you probably already know, whereas some of the biology stuff, I'll be giving you a general overview, but you may be have more questions. And I welcome you to interrupt me when that happens. So I'm going to be talking today about medical and genomic privacy, some challenges, and some attempts at partial solutions. So I know this is the privacy engineering seminar, so I assume most of you all already know the difference between privacy and security. I'm just going to go ahead and make sure we're on the same page. When I talk about privacy, I talk about norms relating to who should be accessing and altering information. Whereas when I'm talking about security, I'm going to be talking about how you implement the choices you may make for who should be accessing information. Okay, so pretty standard. So, for example, if you're dealing with banking records, privacy questions would be who should have access to your credit card bills, maybe your bank, your employer, should your parents have access to it? On the other hand, the security question is how do you ensure that only the people you want to have access to your credit card bill can? Well, there are security mechanisms. My favorite one is envelopes. Those have been around for a really long time. They're really good at preventing people from seeing stuff that they're not supposed to until they open them. But you can also use encryption. And also there are all these legal mechanisms for security as well, which I'm going to be pointing out throughout this talk because it turns out that, at least from my perspective as someone who works in medical and genomic privacy, my primary competitor isn't other privacy people. My primary competitor is the lawyers. Because it turns out the lawyers have spent a really large amount of time figuring out ways of preventing bad people from doing stuff. And it turns out they're really good at it. In some sense, a lot of the more CSE technologies are a little bit more nascent than the various data use agreements, the various legal arrangements that the lawyers have managed to come up with. Okay, any questions about that to start? Okay, so of course, data is power. Many people would agree that, or actually, let me just, a quick show of hands. Who thinks here that records of things you own should not be made public?
That's not me, I suppose.
Okay, so who thinks they should be made public? Things you own should be made accessible to people. Okay, it's always interesting whenever I ask that question because it depends on the audience. Some people actually have no trouble with letting everyone know what they possess. I mean, isn't that sort of the point of social media? You post on something and you show people, oh, hey, I own X. And of course, there was an article in the Wall Street Journal last year about tech leaders behind a plan to build a new city near the California Air Force Base. And somehow we have decided that this is in the public interest. Even though that's private people owning things, we have decided that what land ownership should be one of these things that should be made available. Similarly, there are questions of whether ownership of handguns is something that should be in the public interest. Some people here they say they are. Some people here might be like, no, that's private property. Like, it should have no bearing on whether or not it's in the public interest. So there are all these questions about who ought to own stuff. And the problem, of course, as many of you will probably already know, that many different parties have interests in accessing data or in preventing data from being accessed. And each society in general, and there are many societies on Earth, have to decide as a question of shared values, who should have access to what data. This is not a technical question. This cannot be solved. You can't write down a proof for this. This is something you just have to decide as a matter of norm setting. Whereas security, things like legal controls, those are just implementation questions. You have things like data use agreements. I care a lot about the laws surrounding medical and genomic data, things like HIPAA, GDPR in Europe, the Genomic Information Non-Discrimination Act, which is specifically about what you can do with genomic data. It's not even necessarily who accesses it, but like what are you allowed to do with it. So for example, you may not be allowed to deny someone health insurance because of their genome in the United States, but then it may not be true in China or in India or in Canada, depending on the specific regulatory regimes in those areas. We also have things like informed consent and institutional review boards. On the technical side, we have ACLs, cryptography, de-identification, synthetic data. I imagine that all you all are pretty familiar with these technologies, or at least have heard of them, given that you're in the program you're in. Okay, so now, for me, as someone who works on medical and genomic data, there are certain types of data that are important for us. There's the usual data that people talk about all the time whenever you talk about privacy, your social security number, your name, address, phone number, these sorts of personally identifiable information. Somehow we think of that, or we've generally agreed, at least here in the US, that these are things that should not just be published on a big database everywhere. We are unhappy when it turns out 100 million social security numbers were breached by one of the big credit reporting firms. But that makes us unhappy. Why does it make us unhappy? Well, that's a question of norms, right? So these are general things, and these apply as much to your banking records as they do to your medical records. But when it comes to medical records, there are other types of things. So for example, you have clinical diagnosis like hypertension or COVID. Are these things that should be kept private? If you have COVID, is that something that is something you should be sharing with all your friends? Or is that something that you should be keeping quiet about? Who here thinks that you should share the fact that you have COVID with all your friends? Who here thinks that it should be something that is just kept on the down low, where it's a medical diagnosis? It's just like breaking your leg, right? Well, I mean, breaking your leg might be a bit obvious, but like if you encounter some injury, if you have high blood pressure, is that something you want every one of your friends and family knowing? And so like for different aspects of medical diagnoses, you may have different opinions about whether or not it should be shared. And for COVID in particular, since that's a pretty hot button issue over the last couple of years, there are people who think that that should be made publicly accessible. And lastly, so this gets really complicated because we also have individual level genomes or genome type information. So I work a lot on genomic data. Who here thinks you own your genome? Or sorry, who here thinks you should own your genome? Okay. Does anyone know what that means? What would it mean for you to own your genome? Any thoughts? Yeah, go ahead.
To own the specifically identifying data within your DNA?
Okay, that's a really great start. So specifically identifying data. So you're trying to apply the same sort of idea of personally identifiable information to your genomic data. Okay, so that gets you part of the way there. Who here thinks your parent should own your DNA, or should own their genome?
Their genome?
Yeah. Who thinks your parent should own their genome? I mean, it should hopefully be everyone who just raised their hands a moment ago, if you're being self-consistent at least. But now the question is, can both of you fully own your genomes? And this is one of the areas where this gets a little tricky, because it turns out, like, unlike your banking records or even like whether or not you broke your leg, genomes aren't entirely yours. So they identify you, but at the same time, they're highly shared with a ton of other people. And this turns out to wreak havoc with a lot of the sort of assumptions we have about privacy, because if it's not clear who owns the data, how do you decide who should be able to see it and who has the right to make it accessible? Like, if I were to go out there and take my credit card bill and throw it out there into the world, well, I mean, that is my right. Like, I can just throw my credit card bill anywhere I want because it belongs to me. If I were to do the same thing with my genome, well, maybe my brother would get irritated. I don't know. But because, like, if I share my genome, I'm actually sharing half of my brother's genome as well. Okay, so I will come back to this slide every so often. It's just talking about some of the different ways that different types of settings in which people might care about privacy and security when it comes to genomic data privacy. So there are things, and the issue is that, of course, there are lots of different people with an interest in genomics. You have, for example, a hospital. So that you might be saying, okay, well, you go in, it's very sad, you got a cancer diagnosis, and you want to know what kind of cancer you have. And it turns out that there are genomic tests you can do for figuring out what kind of cancer you have, and those sorts of genetic tests may be helpful in deciding what the right treatment for you is. Now, you may say, okay, well, I just had that genomic test. I don't want that used for anything other than deciding how to treat me. But there are a lot of other people at the hospital who might have other interests as well. For example, they might be like, well, it might be a researcher like myself. We might go in and be like, well, if I happen to know all the different patients who have this particular mutation for breast cancer, maybe I can do a bigger study that will help other patients down the line. And so you have all these different questions of like who is allowed to use your data. And so some fraction of you might say, okay, well, it's all right if you use my data for research purposes for the betterment of mankind. Okay, well, but what really is the betterment of mankind? Like, should a health insurance company might be able to use that information and have more efficient pricing mechanisms for health insurance for everyone? Is that also for the betterment of mankind? And one of the issues we run into is when you're trying to, so people talk a lot about informed consent. Like you want to, if someone releases their data, they should understand what their data is being used for, right? Unfortunately, it turns out that it's really complicated to know all the different ways your data can be used. And when you're talking about, okay, I'll make it available for research, most people don't know all the different wide variety of research that is out there, and which may have other bearings on their lives. And so there are a bunch of different uses you can use for this, as well as different protection mechanisms, ranging from the legal to the sort of more, I'm going to call it technical, though it's not really the best term for it, but sort of making use of CS technologies like differential privacy or homomorphic encryption or things of that sort. And then you also have data perturbation. So this in data perturbation, I'm including things like adding noise for differential privacy or as well as synthetic data if you generate those kinds of data sets. Okay, I've been talking at 1 million words a minute. This, I think, is a good spot to ask if anyone has any questions. Okay, so hopefully you all already know that data is in some sense equal to power, and maybe you can use it for good. Of course, if you can use it for good, you can probably also use it for not good. Let's talk about some of the ways that this is good, and some of the things we want to be able to do with genomic and medical data that most people would agree is for the good of society. So one of the big things that people have been doing in genomics is these things called genome-wide association studies. So some of you might have done a DNA test at some point, and it might have been like, oh, you have a 20% increased risk for heart disease. That kind of data comes from these genome-wide association studies. So basically what you're doing is you're saying, oh, well, by looking at your genome, I can correlate your genome with all these other genomes of people who've had heart disease and tell you that you have an increased risk. And this can be incredibly powerful because it can give you an early warning sign. It can tell you, oh, well, you might want to change your behavior because you are at a higher risk of some particular disease that is manageable or even preventable if you stop eating red meat or something like that. But you notice that what we're doing here is you are precisely associating individual identifiable information with a prediction of health outcomes for an entire population. And so somehow there's an issue here of whether or not that information is something you want accessible. In fact, you're sharing two different bits of information. You're sharing the personal identifiable information of your genome, as well as your medical diagnoses with researchers to be able to build these things. Okay, many people like knowing where they come from. So the idea of tracing ancestry, that's possible using genomes, but only if other people share their genomes. And so you can figure out whether you have 50% Irish descent by whether or not the particular things that identify you happen to be shared with the things that identify other people of Irish descent. Okay, and going a little bit further, tracking criminals by DNA testing may create a safer society. or identifying the remains of victims of massacres. These are things that we might agree are good, or you might not. And lastly, so a lot of those things were more on the genomic side, and we're coming back to the COVID side. It turns out that knowing that a friend has COVID may allow you to prevent the spread of a disease, and some of us would agree that that is good in civil society to be able to prevent the spread of pandemics. We have decided as a society that we're actually willing to incur quite a bit of cost to prevent the spread of COVID. But of course, in order to do that, you have to give up medical privacy. You have to give up keeping private the fact that you happen to have a disease. And there are a number of diseases for which this might be true. Okay, and there are a bunch of dangers. So the sort of basic one is the re-identification problem. So this one you've probably already encountered in your privacy engineering master's program. Well, you may want to be able to figure out that some person participated in the study. Because maybe like the study is correlated with whether or not you're a smoker or some other fact of interest. So there's a re-identification problem. And it turns out that in the early days, at least, so by early days, I mean like just over 10 plus years ago, people were putting information on these public, the Personal Genome Project databases, and they were providing some amount of like demographic information because like in order to do anything useful, you don't like just having the genome is not useful. You need to have it associated with some traits. Well, unfortunately, it turns out that it doesn't take very many traits in order to fully re-identify someone, right? And so Sweeney et al. Were able to link demographics to public records like voting lists and mining for names in hidden attached documents. So it turns out that people weren't exactly careful with posting their information onto this giant genomic database. And if you write a document in Word, it might include your name in it, hidden in the document, and all kinds of metadata like that. And so this is all pretty standard re-identification. This is standard privacy engineering. Like, it turns out it's hard to release data safely. And it turns out that, well, 87% of the US population is uniquely identified by just 3 items of information. So if you know the date of birth, the gender, and the postal code, that gives up the game for 87% of the US population. Okay. Now at this point, I am going to, so this is all standard general privacy. I'm going to talk about some things that are genomics specific, since of course that's why I'm here. And in order to do that, I need to give some general genomics background. How many of you all have had any sort of genomics background in the recent past? So does everyone know what a genome is? Okay, a couple nods. So, biology lesson. The human genome is made up of 23 chromosomes. They come in pairs. Well, most of them come in pairs. And then, of course, you have your XY chromosomes. So those don't come in pairs, not typically, at least. So we have all these chromosomes, and each of these chromosomes you can think of as a long string of letters. So this chromosome 1 is about 250 million letters long. Chromosome 22 is about 50 million letters long. And these are letters in the alphabet ACGT. Okay? And this is what we think of when we think of genomes, because these are the strings that define who we are genetically. And but it turns out that your genome is pretty similar to everyone else's genome, because that's part of what makes you human and not a chimpanzee or a monkey. And so a lot of these letters are shared between, everyone. And so this is how people were able to come up with the Human Genome Project. It was made up of a bunch of, it's not the genome of any single individual, but it was made up of a bunch of volunteers who gave their genomes to science back in 1997. Here's a copy of the ad that was originally released in the newspaper back when people still answered ads in the newspaper to release their full genomic information to the public. So I won't go through the details about all the history surrounding the Genome Project, but basically, because our genomes are all similar, we can create a reference genome and then talk about everyone's deviations from that reference genome. So yes, you could go in and get your genome sequenced and read every single one of the 3 billion letters, but that is very expensive. It is doable, but very expensive. On the other hand, you can just you can read the letters and try to match them to a reference genome and see where you deviate from that, and that is substantially cheaper. And there are a number of these genomic analysis pipelines. So you have your biological data. So maybe that's a cheek swab or a strand of hair with the root attached. And one of the ways of analyzing it is this thing called genomic sequencing, which allows you to read the letters from these long strings. Unfortunately, 250 million letters long is a long string to read all in one go, and we don't have the capability of doing that. So instead, what we do is we read it in small chunks, typically 100 to 1,000 letters at a time, and then we use those small chunks to then map it down to the reference. And so you're like, okay, well, this chunk looks like this part of the reference, but has this small difference. And so that is the basic idea about how genome sequencing and genotyping work. Is you read these small chunks, small substrings of this big string with some deviations, and then you map those small substrings to the original string, not to the reference string, and you figure out how they deviate. And those deviations are, roughly speaking, what make you you. And of course, then you might say, okay, now we have all these deviations. You can then do some tertiary processing statistics to answer biological questions. Does this gene code for brown hair? Does this gene code for blue eyes? Well, that sort of thing. So given aligned reads to the reference genome, so you have all these substrains, you can figure out if a particular position is a variant. And whenever you're talking about protecting genotyping information, normally it is a list of all different places where you might be different. So in this particular case, I have 6 different reads over the same sample area. So these all align to this part of the genome. And there might be sequencing errors. Our equipment isn't perfect. But you might say that, well, if you see something many, many times that is different from the reference, then maybe that means that that is an actual variant. Okay. So that's one way of doing it, which is to do this genome sequencing process. Another way of doing it is there are other techniques that just immediately, they make use of some clever base pairing chemistry. So you may have heard that in genomics, your DNA is double-stranded, A is paired to T, C is paired to G. And we can also make use of that and just probe specific locations in your genome. So you can say things like, I have a probe, it just automatically only attaches to this region of your genome. And if it attaches, then that means you have a particular variant there. Okay, so these are the different types of data, and you can put these on the chip, and then you just get different hybridization areas. So you get a signal for whether or not you have a particular variant in your genome at a particular location. This is the form of the data. And the reason we need to do this is, of course, because genomes undergo a process of mutations. These are typos, effectively. So maybe so our equipment creates typos because we might misread an A as a C, but also your body's equipment for copying genomes also makes typos. And those typos turn into the genetic variation that makes us who we are. So they typically occur roughly once every 100,000 letters during the cell copying process. And some of these mutations are lucky enough to get inherited by your offspring, or that you inherit from your ancestor if they happen to be in your germline. And so this is the source of brand new sequence, and there are many different types of genetic variation. They range from the really short ones, so these single nucleotide polymorphisms, to slightly bigger ones, these repeat regions, which you may have heard. If you've ever heard of like telomeres and longevity studies, like that's a repeat region of your DNA, which when it gets too short, you get old, roughly speaking. And of course, you have entire chromosomal duplications. And so I'm giving you this taxonomy because these will play a role in the different ways that it's possible to use genomic data to re-identify you. Okay, so we have your short things. These are just single nucleotide changes. You can also have insertions and deletions. So this is in an edit distance Levenstein edit distance space. And you can also have multiple length deletions or insertions or duplications. One of these common things that is going to play a role is these things called short tandem repeats. And we're going to see this in a moment because it turns out that when you have a region of your genome that is super redundant, that can really confuse your DNA replication machinery. And so if you have a big stretch, because if your DNA replication machine gets lost a bit, it might be like, oh, I forget where I was. It can't see where it was because you just have this long stretch of repeated regions. And so these repeat regions tend to be a little bit more varying. So like in one person, it'll be slightly longer, in another person, it'll be slightly shorter, just because when it was being duplicated, it wasn't duplicated perfectly. And these signatures can help identify a person because, of course, they're different for each of us. Okay. Like I said, I'm going through this pretty quickly because you don't actually care about the genomics here, but these are the types of variation that can play a role. There are also much bigger deletions, insertions, duplications, inversions, and all that jazz. Just all different ways you can think of large-scale chromosomal changes. Those can happen and do happen. Okay. So we have all this biology that happens, all these mutations that happen to create different strings for everyone. These uniquely identifiable fingerprints are created by this sort of generative mutational process. And so you go back, and this is what creates all the chromosomes we have. And you might notice, though, I've been talking about strings as a single string of 3 billion letters. But we actually have 2 copies of our string of 3 billion letters, or of each of our chromosomes. And so when you're talking about variants, you actually have multiple, you have 2 copies of everything. And so whenever you do this sort of sequencing, it'll often say, well, in this position, you have both an A and a T. Or in this position, you have both the G and the C. And this is because you have one copy that has an A layer and another copy that has a G layer. Or one copy that has a T layer and another copy that has a C layer. But you don't know which of these 3 haplotypes might be present without doing more work. And this is one of the questions that people have been trying to study for decades now is, well, how do you figure out what you actually have on each of your copies of your chromosome? And I'm going into a little bit more in depth in this sort of haplotyping problem, because it turns out that what you are trying to do here is you're trying to extract entire genomes from small amounts of information. And this is great. As a biologist, I'm like, great, you can extract the entire genome from just a few locations in the genome. Now, hopefully, as privacy people, you're thinking now, oh, wait a moment. You can extract the entire genome from a few locations? That's actually really scary. But it turns out that many of the things that are scary from a privacy perspective, for me as a computational biologist, I'm like, great, I don't need very much information. I just need a few bits, and then I get the entire kingdom. OK, so how do we do that? Well, we have all these potential haplotypes because you don't know which of these A's goes with G and which T's goes with C. You can do this individually. So you can there's some process in which you can stitch together individual reads. We're not interested in that right now. The more interesting question is at a population scale, because we're interested in society, right? And society has lots of genomes, and we can use all that extra side channel information to figure out stuff about the genome that we're interested in studying. OK, so what happens when you have a genome? Well, how do mutations occur? You have a mutation, and then it might get passed along. And so, and then you have another process, this process called recombination, where you take 2 copies of the same chromosome, and then you basically mix them. So you swap them. This is part of where our variation comes from. You mix together. And so this is why, if you think about it, if you only had 23 chromosomes and you didn't do any swapping, then basically one of your chromosomes would be from your father and the other would be from your mother. And there wouldn't be any sort of interesting variation. And so this process of recombination is part of what creates interesting variation. I will skip over the mechanisms for this, but just assume that things can swap. It is rare, but it can swap. OK, so recombination. If you didn't have any recombination, you have a mother with 2 chromosomes, a father with 2 chromosomes. I've just color-coded them for ease. The offspring would just have 1 from each. And this would be awfully uninteresting from my perspective as a biologist. And also, it would also be really bad from a privacy perspective, because then you really wouldn't own your genome, because you would be exactly 1/2 of your parents' genomes. So with recombination, what ends up happening is that the chromosomes mix together before you inherit them. And so this creates more variation and also creates some more identifying features, basically, right? Because recombination is an event that happens for each individual. And these shape the genomic structure overall. So if you had some ancestral population of these different colored chromosomes, their offspring would mix these together. And then after many generations, your modern chromosomes, the ones that we see, can be thought of as a mosaic of all these different original ancestral chromosomes. And of course, you might have mutations, and these get mixed throughout the population along with this recombination process. It turns out, actually, that, and this is a question for the statistical geneticists, the majority of human variation is due to mutations that have happened only once and then sort of been passed along if they've been helpful. But the Y chromosome is special. The Y chromosome doesn't come in pairs. So most people who have a Y chromosome only have 1 Y chromosome. And that means that you don't have this whole recombination process, right? So if you don't have a recombination process, what is the Y chromosome associated with? Yes, but also if you have a Y chromosome, your Y chromosome is basically identical to your father's. And this is great because it allows us to track ancestry. What else does it allow us to track? Well, it turns out it allows us to track last names. So from your genome, for the study only did a partial identification, but they were able to just, this is a Y chromosome short tandem repeat. So this is the repeated regions that expand or contract. Scientists were able, this is Melissa Gimreck, who is now at UCSD, was able to identify some personal genomes, identify surnames, link surnames to the personal genomes that people had published, because it turns out that if you just look at the Y chromosome, happens to be associated with fathers, since it's passed down patrilineally. And this allows you to infer other things that are passed down patrilineally, which at least in the United States, has historically been things like last names. And so you can often infer otherwise personally identifiable information from just looking at your genome, because it is correlated with so many different things. And this is super cool from a computational biology perspective, and super scary as a privacy person. If you are female, you don't get a way either, because although you don't have a Y chromosome, you have mitochondrial DNA, which also gets passed down in much the same way matrilineally. Because it happens that we've passed down last names patrilineally instead of matrilineally, there isn't as much of an easy association. But it is the case that if you have a family tree, you can use mitochondrial DNA to figure out your maternal lines as well. Okay, so this is all well and good, but this is only for your one chromosome that doesn't get recombined. So you might say, okay, well, if you're interested in preserving privacy, well, what if we just throw out the Y chromosome and not worry about it? Well, there are several issues with that. One is it turns out that the Y chromosome does some things that some people would consider important, and you may want to preserve those associations when you do a later study on it. The other issue is this doesn't entirely solve the issue, because although this is the easiest correlation to find, there are all kinds of other correlations hidden in your genome. And the tension is that these haplotypes are shared. So remember, I said these mutations get mixed throughout the population, but let's say you are this person here with this blue stretch of chromosome. This blue stretch of chromosome is more or less the same as this smaller blue stretch of chromosome in some other person. And in fact, if you're looking at you and your brothers and sisters, basically half of your genome is shared between you and a brother or sister. It's even about, do we have any identical twins here? No, okay. It's even worse if you're an identical twin, because, well, as much as you own your genome, so does your identical twin. Okay, so the tension is that these haplotypes are shared. Maybe your bank records belong to you. I mean, you may disagree on that. Does anyone disagree that your bank records belong to you? Okay, well, I mean, that's a question for another day, whether or not you actually own bank records, because, also, you don't actually own your bank records in the United States. But, maybe you feel some moral obligation to own your bank records. You ought to own your bank records, even if you don't actually. But and maybe even medical records, whether or not you've ever broken a leg. That is something personal to you. That's not something you share with your friends. Just because you've broken your leg doesn't mean all your friends are going to go out and break their legs, or I hope not, at least. But what about your genome? And as I've been talking about, your genome is simultaneously unique to you. It exactly identifies you, while also being shared with every single one of your relatives. And by relatives, I'm broadly defining that to include the human race. So how do you reconcile the tension? Well, I don't know, and I would love if other people have ideas. This is an active area of research. How do you resolve this tension of privacy around some object that is both entirely identifiable, but also completely shared? Okay, and so let's just talk about one of the cool ways biologists have made use of this fact. And this is genotype imputation. So you remember how I mentioned that often people only care about these lists of these variants, right? So there are the particular so you have 3 billion letters. Most of those 3 billion letters are exactly the same for all of us. A few of them are different. And so whenever you're dealing with anything related to humans, you don't actually care about all 3 billion letters. You only care about the ones that are different. And you can code those in the matrix of whether or not you have a particular variant at a location. And it turns out that sometimes you might not code for every location. Because it turns out that if you're using, say, the hybridization method, each location costs a little bit more money to look at. But all of us are correlated, right? So can you, from just a few locations in your genome, so if you have these locations where you know the answer, so this is just some big data matrix, well, what we're going to do is we're going to try to fill in all the question marks. And this turns out to be actually really, really easy. Because what you can do is you can just create a set of reference haplotypes. So these are the known types of genetic variation. So these are the ancestral haplotypes, roughly speaking. And what you can do is you can just take each of these samples and you can model it using a hidden Markov model and basically be like, oh, well, which of these these reference haplotypes is this stretch of genome from? Maybe it's from this red genome. And if it's from this red genome, because it happens to match this red genome more than anything else in that particular stretch, then you automatically know that all the other locations in that correspond to the red genome. I think in my picture, I actually messed up some of these correlations. But regardless, the references aren't actually correct. But the point is that you can figure these things out because of all these correlations from just a few locations. And so if you gave me a SNP panel of 1 million of your 3 billion locations, I can basically completely identify your genome with, some error, obviously. Even if you only gave me 1,000, that's enough to fully identify you as a person, and as well as give some number of these correlations. And so you don't need very much information to figure out the rest because all this information is shared between the human race. And so you can take small data and turn it into big data. This is great. This means that our studies are 100 times cheaper when you're dealing with GWAS studies, when you're trying to figure out ancestry, than they would be otherwise. But it's also bad because revealing just a few SNPs from genealogical studies, SNPs that by themselves don't talk about disease. So like when you have a variant, it might code for some disease phenotype. It might code for some disease or it might not. But the problem is, even if you reveal a SNP that isn't correlated with, that isn't actually related to the disease, it might be correlated with a SNP that is. So the Genetic Information Non-Discrimination Act of 2008 only prohibits genetic discrimination for health insurance and employment for asymptomatic individuals. There are many other things that people might want to use your genome for. And so, there's also no protection for life and long-term disability, though there are other laws like the ADA sometimes apply depending on the specific setting. So hopefully I have convinced you that genomic data is scary. That's part of the point of this part of the talk. And you can use it in all kinds of really clever ways. And depending on who you are, you might consider this a good or a bad thing. So this is the Golden State Killer. Some of you may have read about this before in the news. This also might be the sort of thing that comes up if you're interested in privacy, because it turns out that there was a murderer who went uncaught for years and years and years. We had genetic testing, but their data wasn't in any of the databases. And so since like the 90s, we've been able to match genomes, but if your genome isn't in that database, you can't match it. Well, but then the police were like, oh, wait a moment. We have all these great databases out there that are just publicly available because everyone wants to find their distant cousins. What if we just find a distant cousin of the DNA sample that they had on file? And they did. So they took the DNA sample they had on file, they sequenced it. The tissue sample they had on file, they sequenced it, and then they were like, okay, well, let's try to find this person's cousin. And they were successful at doing that. So this didn't use the traditional FBI DNA database, instead used a public GED match ancestry website to allow distant relatives to narrow down search results. So this is a website that allows people to upload their DNA because it's your DNA, right? You can do whatever you want with it. And so you throw it up there and you're like, do I have any cousins? Unfortunately, sometimes you do, and sometimes you're the Golden State Killer. And you might, some people here will be like, this is great. Law enforcement has another tool that we can use to track down people who are committing heinous crimes. On the other hand, you might be like, wait a moment. That means that law enforcement can track down anyone. This is, of course, one of the tensions of privacy while you're living in any kind of society. And there are other issues. So, for example, even if the GED match website says that they're not going to share it with law enforcement, which they did after a while. So there was a whole big hullabaloo, and then they're like, oh, we'll only do it in cases of rape or murder. And then they made an exception for an assault case. And so there's this whole big question about, like, what is the website's duty to you as someone who's uploaded data? Once you've uploaded this data, is it theirs to do with that, they please? This is actually coming out right now-ish. There's stuff going on with ownership of these companies, and even if a company currently says they're not going to do anything, the fact that you've given them your DNA means that they could change their policy one day. And unlike other kinds of data, so it's bad if your social security number gets compromised, right? Because then it's this whole paying. People might kind of steal your identity. But in the worst case, you can get a new social security number. It's bad if your password gets compromised. This is so irritating when you get the go on to have I been pwned, and the answer is yes. But you can change your passwords. At the moment, it is exceedingly difficult to change your genome. I don't want to say it's impossible because that's not exactly true, but it is exceedingly difficult to change your genome, and you're basically stuck with it. So once that gets out there, it's out there. Okay, so now we've talked a little bit about that. Let's talk a little bit about more common attacks in the context of these kinds of things. So for example, membership inference. So hopefully this is an attack that all you all have already encountered at some point in this program. If not, I'm sure you're going to be talking about membership inference at some point. So in the context of medical studies, sometimes just the fact that you participate in some study itself might be sensitive information. For example, if you participate in the GWAS that happens to be targeting individuals who are autistic, then the simple fact that you participated in this broad study might reveal a bit of sensitive information. And of course, GWAS only typically publishes summary statistics, not individual level data. But if you've studied any differential privacy at all, you probably know that summary statistics can reveal membership if you do your summary statistics badly and don't add sufficient right amounts of noise, etc. Yeah, that's what I just said. So for example, if the summary statistics suggest that Ashkenazi Jewish lineage is correlated with disease, and the study was done in a place with a very small Jewish population, which isn't Pittsburgh, so I should change my example. But if it's done in a place with a very small Jewish population, then that might just reveal that you happen to participate in that study. Okay, so one of these various other dangers here is study volunteer privacy. So membership inference leaks just a basic amount of information, a single bit on participation. Well, what can you do with a single bit anyway? It turns out, well, there are other types of leakages that are even scarier than like the sort of basic attack on membership inference. Well, much more genomic information can be leaked as well. And as you said, genomic information is kind of scary. And so remember I talked about these haplotype panels, right? So people figured out these sort of reference haplotypes, and that allows researchers to get so much more power out of their studies because they just need to look at a few locations and they can use these big reference panels of haplotypes to then figure out full genomes to do correlations on. Unfortunately, where did those haplotypes come from in the first place? Someone gave their information to this to a research consortium and was like, hey, I want to help researchers. And many of us do want to help researchers. I want to help researchers, and part of helping researchers is creating these haplotype panels so that researchers can study thousands of individuals without having to do a full genomic sequencing. Can we protect those haplotypes? So, can we protect those brave individuals who have donated their genome to science? Well, I mean, originally people were like, okay, yeah, sure. We're not going to reveal these whole haplotypes. Instead, we're just going to provide a web service. It's going to be an online web service, and we're going to say, well, we're not going to give researchers any access to their underlying data. Instead, we're just going to allow researchers to send queries to this database, and we're going to give them some results based on this haplotyping, and then they can use that. And so this way, the researchers never get access to the underlying stuff. Anyone see any problems with that? So it turns out with repeated queries, you can reconstruct the entire original database precisely through the imputation process we just talked about. And so all you have to do is you just send a couple of queries that are pretty much blank, and the imputation server will fill in the rest. What it fills in will look pretty much like the original reference panel of individuals that donated their information, which makes sense, right? Because you're trying to find the nearest thing in the reference panel to what you care about. Yeah, I'm not going to go into the technical details. And so there's this underlying tension of privacy versus utility, which is that many of the questions that we care about answering in medicine and genomics are exactly use the type of information that you may want to care about not revealing from a privacy perspective. So no matter what protections you might want to put on the data, simply knowing the answers to the queries you care about might reveal the private information, because in some cases, privacy is sort of secondary. Like what you actually care about whenever you're playing an online video game is not in tension with preserving the fact that preserving your identity from being known to everyone else on the game. Like those aren't in direct conflict. But in many cases for genomic and medical data, it is precisely the information that identifies you that is helpful to researchers. Okay, and let me give another example of this. So for digital contact tracing, so maybe some of you have had COVID at some point or another, and it's helpful if you can trace whenever people had COVID early, so they can know to isolate, so you don't have a pandemic spread. So that would be really silly for humanity to allow a giant pandemic to spread over the course of a few months that was completely stoppable early on. But anyway, so you might want to have digital contact tracing. So for a while, some of you may even have had a digital contact tracing app on your phone, where your phone is like, okay, well, in order to do digital contact tracing, you need to have a history of the people you've encountered in the past, right? That's sort of the whole point of this, is once you've discovered that you have COVID, you isolate, but then you also have to tell all the people who you had lunch with the day before, or stood in line next to in some big crowded room. And this is hard because we don't always remember everyone who we've ever encountered, especially if we're in a big city and you're encountering people all the time. But now, the problem is, suppose there is any mechanism you have to go back and retroactively tell people that you had COVID. Well, there are a whole bunch of different threat models. One of these is, for example, should the hotel you're living in know that you had COVID? Maybe, maybe not. But regardless, they certainly can if you have any kind of digital contact tracing system set up, because, of course, they could just put another smartphone in every one of the rooms, and whatever mechanism you use to give an exposure notification, they'll know which room had it. And there are, you don't even need a smartphone in every room. You could do some sort of binary search. There are ways of powering up these sorts of attacks. But the basic point is, no matter what mechanism you use, no matter what protections you put on it, assuming you have the answer that tells you everything you need to know anyway, because the thing you're trying to protect, the medical diagnosis, is also the thing you're trying to share, which is the medical diagnosis. How am I doing on time?
So we technically have the room until 50, so I think I usually try to wrap up by like 1:30.
Okay, great. So I will skip over some of these sort of more technical details. And of course, and just talk about some of the general strategies people have been trying to do. So now I've hopefully scared you about things that are difficult. So data perturbation. Hopefully you all have looked a little bit about this. So obviously you should remove names and other identifiers from data that you're releasing to the public. You also might need to remove other sensitive data. And this is where things get a little bit harder. So for example, if you have some rare variant that only belongs to you and 5 other people in the world, then revealing that variant almost identifies you. And so there are a number of different ways people try to protect these. One of them is this idea of k-anonymity, which is a fairly weak notion of privacy, but it's just the notion that, well, so long as k other people also have the same trait, maybe you're not as in danger of being re-identified. Of course, this can be broken with all sorts of linkage attacks, but this is one of the standard ways that your privacy is protected in a healthcare setting. Of course, you can also add noise to the original data or to the released statistics, either using something like randomized response or differential privacy. Normally I go into this in much more depth, but you all have had differential privacy, right? Hello. Okay, so you know the rough story. So the basic idea, I'll just mention k-anonymity because this is actually used by many hospital IRBs. If every record in the released data set is equivalent to k-1 other records with the same quasi-identifiers, then individuals have plausible deniability. It does reduce the utility, and if it's done poorly, other weak identifiers can be combined to relink individuals. So this is a major problem with this, but it is sort of the basic notion of privacy that is used often in the field. Okay, and randomized response is a way of plausible deniability by just choosing whether or not you're going to return the right answer. And differential privacy, which is super cool. It's strong, provable privacy that is very rarely used in practice, at least in the medical fields, because it turns out to be hard to work with and hard to explain. But the basic idea is you have neighboring databases, and if you can give plausible deniability to people, to a record having ever been in that database for the outputs of your query. Skipping all that. Another approach is, of course, generating synthetic data that is probabilistically similar to the old data. If the data is still similar enough, you can train models, perform analysis without revealing original patient information. But how similar is similar? And some of you all probably know this better than I do, but it's difficult to quantify how similar a synthetic data set is to the original one and how much information is leaked between them. Well, depending on what sort of protocols you use to generate the data set. Let's see. Okay. And let me just talk a little bit about privacy and utility. It turns out that because the data that we're interested in, your COVID diagnosis, is exactly what you're trying to protect as well, mechanisms like differential privacy are not actually that useful for it because you're precisely putting noise on the query like you're interested in. So you can give a differential privacy guarantee for a digital contact tracing app by just probabilistically choosing whether or not to inform people that they had a contact with COVID. But of course, that reduces the utility of the app quite a bit if you don't always tell people when their friend had COVID. But of course, that is what would be required if you wanted to actually protect the privacy of the friend who had COVID. Okay, so that is all sort of centered around individuals. Another big problem in the sort of medical and genomic space is, of course, for researchers. You might have a lot of different hospitals or a lot of different health insurance companies or drug companies who want to jointly compute on some complicated data set in order to power up their analysis results. But of course, you may not want to share that information with each other. Hospitals might not want to share their full patient medical records with another hospital. Though they might be willing to be given enough legal controls. Regardless, when many parties are collaborating to do an analysis, computing information might inadvertently reveal private information. One solution is legal data use agreements. This is how people actually do it in practice. So the lawyers are beating out the computer scientists substantially as far as I'm concerned in this area right now. So there's a lot of work to do. You can also add noise, but of course this comes at a cost to accuracy. Another approach that some people have been playing with lately is, of course, homomorphic encryption and secure multi-party computation. The basic idea is, for example, have you all done homomorphic encryption or MPC yet? A little bit? Okay, so let's just say that Alice, Bob, and Carol want to know an average weight, but not reveal it to each other. So this is a sort of basic example of secret shares. So Alice has two values, A and S1. Alice sends where A is the weight. So Alice sends A plus S1, which is some random number. Of course, you add that noise to Bob. Bob now knows A plus S1 and B and S2, B being Bob's weight. He sums it all together, sends it to Carol. Carol now knows C and this sum of all these things. Send it back to Alice. And then what you can do is they can remove all the noise, the S1, S2, S3, the secret noise, because in this further protocol. And at the end of the day, we know that the average weight between all three parties is A plus B plus C divided by 3, without any one of the individuals having revealed their individual weight because of these secrets that you've added to it. And so there are more complicated versions of this that allow you to compute statistics that you're generally interested in without the intermediate values revealing anything. You also have things like homomorphic encryption, where you're allowing a central server to operate on encrypted data without revealing the plain text. And this is also really powerful because you get the strong cryptographic guarantees that the intermediate data just looks like noise unless you have the private key for decryption. I will skip the RSA example. And this has been used in a number of different settings, such as GWAS across institutions. So if you have lots of different institutions that all have patient data and they want to do some complicated analysis, joint computation without needing to share everything with each other, then you can do some sort of MPC-like protocol to allow for the sharing to only reveal the final result. This doesn't protect the final result, of course. The final result may still reveal information, as we've talked about, but at least the intermediate stages are guaranteed to not reveal information. And it turns out that for many of these computations, using MPC does not come at a cost, or homomorphic encryption don't come at a cost to accuracy, but they do come at a cost of about 1,000-fold in runtime. Okay, I was going to talk a little bit about some of my work, but I think I'll skip most of it. But basically, we were working on combining probabilistic sketching with homomorphic encryption to allow you to do these sort of combined analyses. So if you want to know how many patients at hospitals 1, 2, and 3 all had hypertension, well, you can't just add the numbers together because you have to deduplicate, because the same patient might be at 2 different hospitals. And so one thing you could do is you just add them up, but then you have an estimate that has a wide range because you don't know how many patients are at all the hospitals. One solution that hospitals have actually used is you just hash the social security numbers. This turns out to have issues, as you might imagine, because yes, we have good cryptographic one-way hash functions. But if you're looking at hashing social security numbers, it turns out that there is a limited range of possible social security numbers, and you can just do a dictionary attack on it. Another method is to use some kind of secret sharing. And so we did we combined some hyperlog sketching with homomorphic encryption. We changed the protocol a little bit to make it work well in the homomorphic sketching framework. And that allowed us to protect privacy that way. I should point out that these sorts of probabilistic sketches don't actually provide privacy, which I think may or may not be something that Damien talks about next week, because as Damien, this funding, said cardinality estimators do not preserve privacy by themselves. This is something that we've also done studies on to show that these sorts of sketching algorithms don't preserve privacy precisely because they allow for deduplication. It turns out that deduplication of a data set is actually the exact opposite of protecting privacy, right? Like if you can deduplicate, you can reveal that someone was in the data set, but if nothing changes when you deduplicate. Okay, and I will skip over the full protocol. And just mention that there are some other techniques people have been playing with. One of them is these trusted execution environments. I don't like them that much because you have to sort of trust Intel, but if you trust Intel, you can use things like Intel SGX, things like that. Trust Intel if you don't trust Amazon when you're doing computations on the cloud. I think of it as just moving the trust to a different giant corporation, but your mileage may vary. Yeah, so just in conclusion, privacy questions, you should have data. Security can be used to implement privacy decisions. The problem is complicated because genomic data is shared and many people have legitimate uses for precisely the sort of thing that you want to protect privately. The problem is that the things we care about are exactly the things people care about from a research and like healthcare perspective are exactly the things we care about from a privacy perspective. And you can use a combination of de-identification, noise addition, and cryptography to try to implement some kind of privacy. But I think as my last thing, I'll just mention that, repeat the thing I've said a couple times, that the lawyers are way ahead of us in that what they've just realized is, well, you don't actually need to provide privacy if you just provide sufficient penalties. Which is the way things like HIPAA work in the US. So HIPAA penalties, so that's the Health and Insurance Portability Accountability Act. HIPAA penalties are actually really substantial because they scale with the number of records that are released. So it's not like certain types of other penalties where you're just like, oh yeah, it's just the cost of doing business. Here, the penalties actually scale in such a way that hospitals are strongly incentivized to not release data. And this isn't to say they're perfect, but it does keep them from normally releasing data. So legal penalties are also useful. And I think I will end there and have time for any questions about any of this.
Thank the speaker. Questions from the...
Yeah. It's the name of your paper. I'd like to.
Oh, yeah. I put it sort of near the end because a lot, federated Boolean queries via homework encryption. Published last year. It's like, if I'm speaking frankly though, like I think there's a really big gap between the sort of technological mechanisms we have and the sort of queries. Like no hospital is going to use this sort of protocol. And I've talked to the hospital systems about this before, like because they're just too complicated. When I worked at Harvard Medical School for my postdoc, and we did some amount of trying to do some amount of privacy protection, but implementing any of these more complicated frameworks requires that the doctors understand what you're doing. And that can be tricky. Like, and there were really brilliant doctors at Harvard Medical School, but like you still need to be able to explain exactly what you're doing and why that gives privacy in a way that the lawyers are just better at than we are. So that's something we need to do a lot more work on. I think this is especially about differential privacy. Someone needs to come up with a really good explainer of differential privacy for like physicians or policy people or whatever, because every time I've tried explaining it, people are like, well, what is this epsilon thing? Like, what does it actually mean? Like, at least with k-anonymity, like I can explain k-anonymity to really anyone in about 30 seconds. Differential privacy takes a lot more work. And I think that's one of the various hindrances for like wide adoption. Question?
Do you think there's a bias towards a particular technique that exists in biocom?
So right now, if you're working in computational privacy for genomics, a lot of people are doing, there are two major approaches. So like there are the people who are doing things like secure GWAS through MPC and homomorphic encryption. And then there are also the people who are trying to do differential privacy on top of that. The differential privacy stuff is a little bit complicated because you have to deal with lots of correlated tuples in your data. So I don't know how much differential privacy you know, but with differential privacy, if your records are highly correlated, differential privacy starts losing some of its guarantees, roughly, right? If you have 10 copies of a record in your database, then you can say, oh, well, any one copy may or may not be there. It doesn't actually help you too much. And because of all the correlations you have in genomic data, this is actually quite tricky. So you'd have to, so there's some work on trying to come up with a decorrelated set, roughly speaking, of genomic data so that you can apply the differential privacy and actually get the guarantees that you're supposed to for with differential privacy. it's also tricky because there's some sense in which there is a ton of side channel information that you have whenever you're working with lists. Basically, any information about any human is extra side channel information you could use to try to break through the privacy of some of these mechanisms. Because we are all correlated genomically. But I would say those are the two major directions people are going right now. In the medical space is basically entirely dominated by lawyers right now. There is so a lot of the big medical research databases for the count queries. Like if you want to ask a hospital how many patients have hypertension, they usually will give you like 437 plus or minus 10. So they do have this sort of they add a little bit of fuzz to the data, but it's not especially. It's systematic, but the fuzz isn't what's actually protecting it. It's just the additional layer on top. And instead, what they actually do is they just keep a log of everyone who has access to the data and every single query that's ever made. And then every so often, they audit it. So those are the mechanisms that are actually used. Question?
23andMe, you mentioned, there is a whole downward spiral that's happening.
Yep.
So should people be worried that, they'll be targeted for like specific diseases?
So it depends on what you're worried about. It is true that right now, health insurance is not allowed to target based on genomic information in the United States. So there are laws surrounding that. But I think life insurance isn't protected, if I remember correctly. So for that, I really think that at that point, the cat's out of the bag. The only solutions we have are legal solutions. One of the things I would think would be useful is if there are enough technological controls that you don't have to do this sort of legal cleaning up the slate after things are already released. I don't know how much people should actually be worried right now. I don't know. Yeah, I wish I had a better answer. Any other questions?
Can I ask a clarifying question about
Yes.
that?
What's up?
So I actually don't know anything about GINA, this law that prohibits discrimination based on your genome. I hope it's something like if you learn about the likelihood that I am going to have a disease versus somebody else is going to have a disease as a result of learning my genetic information. I would hope it also sort of protects the downstream stuff there. So for example, right, let's say I have a 60% chance to have cancer, but so-and-so only has a 20% chance to have cancer. Sure, they can't discriminate based on the genetic information itself. Does that sort of continue to protect you one way down? So if they learn that I have, a 60% likelihood to have cancer from my genes, can they then discriminate on the basis of the fact that I am likelier to have cancer? I hope that made sense. If it didn't,
So
I actually just don't know anything about GINA.
I don't know. I can't give a definitive response. My sense of that is it protects that information from being used to predict your risk of cancer.
Okay.
But they can still discriminate against you based on your risk of cancer via other signals.
Okay.
Because that is certainly allowed. So, sorry, I was trying to find I thought I had a quote from it, but
I'm just curious.
But I haven't.
Because in some other privacy laws, I guess I have noticed. So, for example, over the summer there was a sort of attempt at a Pennsylvania privacy bill.
Yep.
One of the things from that was, I think, biometrics themselves counted as sensitive information, but mathematical representations derived from biometrics do not count as sensitive information, which seems weird to me.
Yeah, I think my understanding is that GINA protects it from being used.
Okay.
So I don't know about how many levels of obfuscation you would need to do. I don't know exactly how it's worded. So yeah.
I'll ask one more.
Yes.
Question. Okay. Some of the stuff you talked about earlier, there's sort of an ownership question and a privacy question.
Yes.
I think a lot of the technical protections we have for privacy sort of assume those things are the same thing. But also, I think, to some degree, you can own something without it being private. I don't want to bring this into it, but intellectual property is something that we're okay with other people, using in certain situations. But I can, if I own the IP to something, I can still control how it's used, at least, even if it's not private or not confidential.
Yes.
And I guess I wonder if you have any thoughts on like whether is it the ownership part of it that's a problem, or the privacy part of it, or is it just a big mess?
My pessimistic view is that genomic privacy cannot exist.
Okay.
So that is my pessimistic view, and like I say this as someone who sort of works in it. But like my pessimistic view is that genomes are so inherently shared between everyone that there isn't really a way of protecting it. Sort of like there are ways you can protect information that is sort of surrounding it, like your participation in the study that reveals your genome or things like that. But like the genomes themselves, I think of as the shared intellectual heritage of humanity, for lack of a better word. And I say this partly also because I don't want people patenting parts of our genome, which people have tried doing at various points. And so like I do think of it as a natural product, for lack of a better phrasing.
That is an interesting, sorry to just keep monopolizing.
Yeah.
That's an interesting question. I do know that some biotech companies patent like bacterial genomes.
Yes.
Which is obviously different than a human genome, but I would imagine there might be some other issues. Does that extend all the way to like E. coli or something?
My personal view, and this is not me speaking as an expert, this is me speaking as a human, is that things like that, I think it is reasonable to patent the use of a particular stretch of genome for a particular purpose. Like patenting a particular gene for the purpose of treating some disease or creating some natural product, I think that's actually pretty reasonable. I don't, because like, you're patenting the use. The invention is the use of it, but not the letters themselves. Yeah.
Any other questions?
Yep.
You believe it's basically public information, or would have to be, specifically because it's shared, or because it's so easy to derive from so little data?
So, I mean, those are related, right? So, part of what's happening right now in the genomic space is lots of countries are building these big national biobanks with lots of genomic information from a representative sample of their population. So like in the US, we have the NIH All of Us program. The UK Biobank has been doing it for years. There's a Japanese biobank. China has some as well. And these big biobanks are storing millions of genomes of humans from different populations, along with some identifying characteristics, because this is incredibly powerful for understanding disease. But with that set of millions of genomes, like the shared nature of the genomes means that it's very likely you're not too far removed from someone who has shared their genome. And if that's the case, then with just a few bits of information, just a very few bits of information, they can probably recover yours as well. And so that's the sense in which I don't think that genomes can really be private in that sense. Yeah, my very pessimistic view. So, yeah. Was there a question over here? Or?
I mean, I was thinking about on the topic of ownership. Like, I read about doing genetic testing over embryos,
Yep.
for children. How does ownership even, for children?
I don't know. I really don't know. Yeah.
I mean, legally even.
I'm sorry. I don't know how to answer that. I wish I did, but that's not something I consider. You need to talk to a lawyer. Is the real answer. Yeah.
How derivable is like a unique identifier like the fingerprint or the iris from a genome? Because ultimately, if the genome isn't protectable, neither is any identifiable figure.
Your fingerprints aren't actually derived from your genome, and I think your iris isn't either. So they're the result of some random process that happens when you're in the womb, basically. And so and so identical twins don't have the same fingerprints. So it's not the only biometric if you're talking about biometrics. It is one that's really hard to change, but that's true of most of your biometrics. The difference is when it comes to a genome versus a fingerprint is that a fingerprint is more strongly yours. It's not shared. I don't know. There probably is some genetic correlation to it, but it is not shared in the same way that your genome is shared.
Any other questions? Okay, let's thank Lena.